Alright, welcome to AP Biology again. I'm glad I got to meet all of you today during class. And this is going to be our first video, so this is really how things are going to go. You'll find my video on YouTube for the next class period, and you'll just watch that after you've taken your notes on your sections. And it'll go really nicely with what you've read, and hopefully it'll just add a little bit more info, maybe more a little bit. I'm trying to hone on the most important things, and it'll help you access what we're doing in class and what we're reading a lot better. Just another way to help learn some content. So I titled this Welcome to AP Biology. Welcome to AP Biology. So I'm glad you're here. I know we're going to have a great year. So the first thing that I'm going to do just really quick today with this quick PowerPoint is a basic intro to the scientific method and experimental design. Both these things are extremely important to AP Biology. Talk about most of your questions, not just on the free response section, but on the multiple choice, will have something to do with experiments or looking at data from an experiment, designing an experiment, procedures, techniques with experiments. So this is extremely important. And I call it AKA Why We Made Baking Soda Volcanoes in 3rd grade, because I know you've probably done that experiment 50,000 times. But the reason your teacher had you do that was to be able to form hypotheses, to test your hypotheses, and to come up with a conclusion. What happens when you mix vinegar and baking soda? Why do we see observation, that bubbling, that overflowing? So, to start with, just a little quick history. These two guys are really important. Plato, right here. So, right next to that, Plato. And over here we've got Aristotle. And these gentlemen had a lot to do with inventing the scientific method. So Plato, Greek philosopher, was very concerned with the metaphysical. Why are we here? Is there a God? What is our purpose? And he basically just thought about things to come up with this revelation. So he didn't really have a process in mind or that he practiced that helped him reveal these truths. It just came to him. Whereas Aristotle kind of represents the new, the scientifically minded, and he used a sequential approach of deduction to be able to learn or discover new information. He is kind of where science begins to diverge from philosophy. Alright, so to be able to do this, to really be able to get into the scientific method in a way that suits AP Biology, I'm going to assume that you already know some things. I'm going to assume you already know what a dependent and an independent variable are. So when you're making an experiment or designing an experiment, what a dependent variable is and an independent variable is. I'm also going to assume that you know what a control group versus an experimental group is in an experiment. So when you're making an experiment, what is the control group and what is the experimental group? I also am assume that you know how to make a basic graph by hand. So not just with Excel, but by hand, pencil and paper. Drawing graphs and interpreting graphs, that is going to be, as Donald Trump says, huge on the AP test. This is going to be huge for the test. Some other things I know: how to write a simple experimental design. So for example, if I say you want to design an experiment to test the hypothesis that plants that are exposed to heavy metal music grow more quickly than plants that are exposed to silence, how to do that? I'm going to assume that you know how to write a basic hypothesis, and I'm going to assume that you know a little bit about the scientific method. So the make observations, experiment, test the experiment, Does it support your hypothesis? Research, things like that. And if anything, any one of these are things like, no, Mr. Munch, really doesn't ring a bell, I'm going to direct you to my A+ session. So please take advantage of my A+ session, specifically the AP Bio ones, in which I'll help you with all of this stuff. And if you had any questions from Chapter 1 or Chapter 2. So for example, if you didn't really remember a lot from chemistry, I would suggest going to my AP session. 4 things that I'm really going to teach you right now. The more fluid or real world, whatever that really means, version of the scientific method that actually fits what the process of science is for laboratory scientists in the real world. We're going to review very briefly some types of scientific variables and the experimental design. And then I'm just going to give you a quick overview of the laboratory write-up. I will be giving you sheets for this and explaining it more next week. Alright, let's get started. So the scientific method, aka making baking soda, volcanoes in 3rd grade. So the scientific method, which is kind of here on the left, is a way that we can go sequentially about discovering the answer to a question. So, for example, like we said, do plants grow faster when exposed to heavy metal music as opposed to silence? And these different steps you should find very familiar. And this, what we probably learned since 3rd grade, works for a lot of cases, but it's extremely simplified. Real science is a lot more cyclical. You go from this part to this part, to procedures, to asking questions, to background, to conclusions, to analyzing other people's data. This line just doesn't cut it. Doesn't really reflect what actually happens in science. It assumes that there's no one else involved in your experiment, that you just do these steps, check, 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 and there's no one really else involved in this whole process with you. Most of the time in real science, if you're if you're doing an experiment, by the end, let's just say that no one does an experiment alone. And it doesn't really address why you're doing the experiment. What's going to come out? What's the benefit? Why is this important? This drawing is a lot more representative of what we're actually going to do in science. It includes the basic scientific method right here, making hypothesis, results, interpreting the data, supporting our hypothesis, opposing, is it going to inspire some new research we're testing? This is the experimental design portion right here in the beginning. This is pretty much what was on the last page, but it has some other sections. There's a community part. Right after you do an experiment, publish a paper, something like that, other scientists are going to try to replicate and review your research. There's going to be huge discussions and lectures and symposiums, if what you've discovered is groundbreaking, of what that means. Your research might lead up to a theory being built, or a large generalized explanation of a natural phenomenon. So the scientific community is going to be in that scientific method process. Also, your experiment or inspiration for your experiment might be rooted in developing new technology or addressing a societal issue. Just building pure research, building knowledge, does it solve an everyday problem? Like Velcro. That's how Velcro was invented. It solved a problem for NASA in space. And so our benefits and outcomes are going to influence the scientific method, as well as making observations and asking questions. And all of this is influenced by kind of the experimenter's personal motivation, new technology, and curiosity, and things like that. So this process, and I'll have a picture like this up in my room, is a lot more reminiscent of what we actually see happen in science. We skip all over from circle to circle, from box to box, until eventually we don't even know what's happening, but we can say we're doing science. Doing science. Not very technical, but it gets to the heart of what we're talking about. Second part about the scientific method, I think there's no better example than Jonas Salk right here. He first developed or discovered antibiotics, penicillin, by working on penicillium bacteria, which he has in these agar plates right there. So it's a great example of real science at work in the scientific method, because through his experimentation, did most of the parts of the scientific method we want to look at. So the story goes is that Jonas Salk had made some bacterial cultures, some penicillium bacteria some bacteria cultures, some staphylococcus bacteria cultures rather, and stacked them all up by the windowsill right here before he went on holiday. Well, he came back on holiday and some fungus, some penicillium fungus were growing in it. And he noticed something really strange, and that really strange thing was right around the mold, no bacteria was growing. No bacteria was growing. And he was like, I wonder what is causing that to happen. So the problem that he had, his experiment, his research is based off is, why isn't the bacteria growing? Why isn't the bacteria growing? And from his work as a microbiologist, his hypothesis, what he thought the explanation was, is that the fungus was releasing some sort of chemical. Some sort of chemical that was killing the bacteria. And now, decades later, we learned that that chemical was the first antibiotic, penicillin. So the things that you can see in his initial experiments was that he formed a hypothesis. Hypotheses are the building blocks of science. They're those testable, proposed explanations for an observation, an educated guess. What do we guess is going on? And it's going to be the prediction that we make for the experiment that we use to test our hypothesis. What's going to happen? So when Alexander Fleming wanted to investigate how the chemical compound the bread mold was secreting affected bacteria cultures, his hypothesis was this chemical will kill the bacteria. Wherever he puts that fungus, and I'll just put the fungus, the fungus's chemical would kill the bacteria. So he's going to set up his experiment to test this. In an experiment, you have an independent variable, and that's the variable that's being manipulated or changed in the experiment. What the scientist is investigating. So he's investigating whether this chemical, this fungus, will kill the bacteria. So the independent variable he's going to use, what he's going to manipulate is the presence of the fungus. So he's either going to have the fungus present on the petri plates, the bacterial cultures with the bacteria, or they'll be absent. That's what he's changing. And by extension, if the fungus are present, so is the chemicals. If they're absent, the chemicals aren't present. The dependent variable then is what changes in response to the independent variable. So what changes depending on if the fungus is there or not. And what changes? What's the dependent variable? Bacterial growth. So if the fungus is there, his hypothesis is that the bacteria will grow. The fungus is absent, if the fungus is present, the bacteria won't grow. They'll be killed. If the fungus is not present, the bacteria will grow. And in designing his experiment, he has to treat with things called confounding variables. So some other variables besides fungus that might have an effect on bacterial growth, such as maybe temperature, or light, or nutrients. And your goal when you're designing an experiment is to keep or to minimize these confounding variables, to make sure it's not these confounding variables that are really affecting the bacteria instead of the fungus or the chemical. So, in making his experiment, Fleming created a control group, or a set of Petri plates, that he never changed the independent variable. So, for example, he swabbed some bacteria on a Petri growth plate, and he then saw the cultures grow, and he applied the fungus that secretes the chemical to the growth plate. And actually, these should be switched. My bad. Experimental group should go with this picture. Control group should go with this picture. My apologies if you're confused on that. But experimental group, where you are manipulating the independent variable, he is applying fungus. After he let them grow, the fungus killed the bacteria, like you can see on this original image of his Petri plate. The control group, he's not applying or changing the experimental or independent variable at all. So he's still growing culture bacteria. He's not applying any fungus. And at the end, he just gets a plate that's full of bacteria, no spots like this from the fungus. This is important because in having a control group, because it gives you a benchmark to compare what happened in your experimental group. If this experimental plate looked just like the control plate, he would know that the fungus really didn't do anything. So he can measure the extent to which the independent variable affects the dependent variable by comparing the experimental and the control groups. And here is actually a test that they do now of a plate filled with bacterial cultures with many different antibiotics, and you can see which ones are more effective for this bacteria, because they will have a larger, more defined circle like this one, instead of smaller, less defined circle like this one of these two. Alright. So, in order to then take their discovery and communicate it with the world, scientists have to write, and written communication is very important in science and in this class. In this class, half of your exam will be based on your ability to communicate things in writing, essays, and long form. Scientific writing happens in published papers, so like Science or Nature magazine, things like that, and in lab reports or lab write-up that we'd use to keep journals, scientific journals. Your writing needs to be concise to the point, supported by data, and get your point across clearly and consistently. On the AP test, they don't care if you write an introductory paragraph. They don't care as much about your spelling, if you indent any of that crap. They just care if you are able to support your opinion, and it's clear, concise, and shows them that you know what you're talking about. So, two types of writing we're going to do a lot in classes. Do a laboratory write-up, or a journal. Looks like this. This is where you document or you keep track of your scientific experiments. Scientists use a journal like this. And it has a uniform kind of setup we'll talk more about next time. A title, a purpose, why you're doing the experiment, your hypothesis, right? What's your prediction for the outcome of your experiment? Like, if I subject a plant to rock and roll or heavy metal music as opposed to silence, then the plant will show more growth as measured by a meter stick than a plant that's not subjected to heavy metal music. What materials did you use? What did you do step by step to the experiment? Your data, what happened? What's your graphs, your numbers, your data tables, and your conclusion? What does this data mean? What went wrong? What should we do next time? So this is what would be in your lab report, which you write in your journal. What ends up getting published in things like Nature magazine are formal lab reports or formal research papers, and they're a little bit more extensive, and we'll be doing 3 or 4 of these, maybe 1 per quarter this year. But this is really how we publish scientific data. You can see we have some similar sections, but also we have like an abstract, an introduction, a works cited, and an appendix. So it takes more time. They're between 5 and 10 pages usually, but they are very detailed, and they are what gets published in scientific journals. Alright, so how does all this relate to what we need to learn for the test and what's important? Well, there's 2 examples of specific AP questions you might see on the exam that have to do with this information. They're not going to ask what's a dependent and what's an independent variable, but they might say, you notice the geraniums in your living room grow much better than the ones in the garage. You wonder if this might be the case because your brother is always playing loud music out in the garage while it's quiet in the house. They design an experiment to test this. So they're testing the hypothesis that sound levels affect plant growth, that plants subjected to a higher decibel level will show less growth than plants that are not. They will then ask you to design an experiment to test this. Maybe it's a free response question. You need to have the experimental design, your materials, your procedure, hypothesis. So they might ask you something like this. They also might give you a graph like this from the data section of an experiment like that and say, what most likely could have been a potential hypothesis for this experiment, or what could be a potential conclusion that you would reach if this graph was presented in a study of the effect of sound volume on plant height. And you'd have to look at this, and you'd have to explain what a potential hypothesis or a potential conclusion would be based on this one piece of data. And we'll get some practice during class, but this is how this all kind of works together. Alright, so that's kind of it for today. I want to make sure that